All right, what we're going to do today is finish up the eukaryotes, and these are some interactions of eukaryotes and other organisms, other eukaryotes. And some of them are maybe not even fully in your notes. You may have to take a couple of extra notes. And some of them are a little bit disgusting, so viewer discretion's advised in this class. I'm showing you some things off your slides that can be a little bit difficult to look at. So some of you, that makes you concerned, be ready to look away on certain slides. But what we're going to do is talk about, quickly, the origin of eukaryote cells. It's basically an endosymbiosis again. How did mitochondria, chloroplasts get in there? We won't spend very long in there because we've been talking about it in past lectures. And then we'll move on to these interactions here that are fantastic. Quick jump into the beginning of viruses, and we'll call it a day at that point. So get your virus notes handy as well. So this is summarizing what is generally known about endosymbiosis. When did the mitochondria and the chloroplasts get inside those cells? When were they enveloped and then used as energy-generating organelles? And so here this is demonstrating that a long time ago, a eukaryote progenitor, so something that's ancestral to what we see today, would have engulfed a bacterial cell and enclosed it within its own membrane as well as a bacterial membrane. Somehow the cell wall is now gone from the bacterium. And this is ultimately the beginning of the mitochondria. And later, it would have been later, as you can see it in the tree, chloroplast engulfed later. So we already know ancestor of the mitochondria. It was in the bacteria, the proteobacteria, and the ancestor of the chloroplast, something like cyanobacteria. But ancestors of the cyanobacteria. Not modern-day cyanobacteria. Okay, so basic tree here, chloroplast started somewhere later, mitochondria somewhat earlier, which is part now of almost all life. So do we have other evidence for this? This is sort of a summary slide, which summarizes for you all of the evidence we have that, yes, the mitochondria, chloroplasts, these are bacterial in origin. And for a while, it was very important to justify this because the data were weak, it was just an idea, and it was very controversial. So it had to be defended with evidence like this. For example, they resemble bacteria in size and shape. They divide like bacteria. And by divide like bacteria, here's our Fit-Z ring again. They use Fit-Z to delineate the center of the cell for cell division. You get that Fit-Z ring, which is a bacterial feature. And you then also have the membranes that I mentioned, where you have both the bacterial membrane still there, and you have another membrane, so it's a double membrane, which reflects the membrane that came from the host itself. So 2 layers of membranes. Not sure what happened to the cell wall in all of this process, but you don't really need it as much in the environment internal to a cell. So each has its own DNA and ribosomal RNA, and you sequence them and you see, wow, it's bacterial sequences here, right in the bacterial portion of the tree. And they have circular chromosomes in contrast to their hosts. All of these, good evidence supporting that these are bacterial in origin. And although we know that there are amitochondriates, they don't have any mitochondria at all, and you should remember Giardia was our example as our model organism from last class. Beaver fever doesn't have mitochondria, it ferments anaerobically instead. So even though they don't have it, you can see evidence in the genome that used to be there. So this is something that really is basal to all eukaryotes. Probably all eukaryotes had mitochondria at one point in time, and the vast majority of them still do. So they probably evolved these Giardia, not needing them anymore where you find them, where they need to live. So then there's also experimental evidence today that this kind of endosymbiosis can still happen. And so this is from, I always like to honor those who came up with the work, Kwang Jian from the University of Tennessee. You don't need to remember who exactly did this experiment. The point is that what he did was to identify that if you take amoeba from the environment and you look at them, you realize that there are bacteria inside of them. And he called them at the time X bacteria, because no idea why they're inside of amoeba and what benefit they have for the amoeba, if they have some benefit. But one thing from the experiment that's shown on this slide, what he figured out is inside these amoeba, living bacteria, and he found that if among those amoeba that don't have any bacteria, you add these same X bacteria to them, some of them will die, but some of them will survive with the bacteria now living inside of them. So maybe a rough start, but some of them end up with X bacteria inside of them. And then here's the key thing, after a few generations, not very long, you treat them with antibiotics, which only affects the bacteria, it does not affect the amoeba, but nonetheless the amoeba die. So those amoeba with X bacteria become dependent on those X bacteria, and you can't get rid of the X bacteria anymore, because they'll just die. So that implies that this endosymbiosis that we see in organelles today can be established in a modern day short time frame and result in a mutual reliance one on the other. Okay. So that's the experiment, and it was actually unknown what benefit there is. Still don't really understand why this dependency occurs. But what occurred in 2004 was someone said, well, what kind of bacteria are these X bacteria? Maybe we could stop calling them that. So they do PCR, they sequence that ribosomal RNA gene, and they see that the X bacteria are actually Legionella, which is a genus of bacterium that you will find is the causative agent of Legionnaire's disease, which we can also contract if we don't keep our hot water heaters turned up high enough in our house. We have to make sure you keep it about 60 degrees Celsius or higher, because that will kill Legionella, or formerly known as X bacteria. And so this is another type of intracellular pathogen which can establish itself as an endosymbiont of amoeba. So that's Legionella. So that's a modern day example of endosymbiosis. And here's another one, and this one's kind of neat. Do you look at Paramecium in 240, L, the lab? Not yet maybe, but I'm not sure if you will, but they're really big, easy to look at, very motile, and they eat everything in sight. And sometimes if you put them in the right environment with the right types of algal cells, as you can see right here for example, they start to swallow up the algae all around them. Eat. But they're not actually eating, they're not digesting them, they're just storing them up inside. So you look under a microscope and you say, hey, I've got an algae here in my culture, but no, it's just Paramecium, a eukaryote, and all it's doing is putting them inside and sort of sucking on them like Jolly Ranchers for a really long time, because while they're photosynthetic, they're also generating organic carbon, and that organic carbon can then feed Paramecium. So you suck on the Jolly Ranchers for a while, but when you get bored of that, you chew them up, you crunch them up, and you swallow them right down. So Paramecium does the same thing. At the end of the day, it gets dark, the algae is not really useful anymore, and so they just digest them all, eat their meal at night as well as throughout the day. And then they'll start again the next day. So that's a modern day example of very much similar endosymbiotic event, which benefits the host and maybe the algae for a few hours. And so these are good examples to demonstrate it can happen. It can happen a lot. We need more examples like this to understand how a long-term organellar endosymbiosis can establish itself. Any questions? Or is that okay? Okay. So here's a few lingering questions. If we can show it occurs in experiments, why are there only 2 organelles that we know of? Why did it only establish itself that permanently twice? And then we know now the ribosomal RNA sequences that are of the organelles, but we don't really know what that would have looked like as a free-living organism at some point in the past. So we don't know a lot about that early organism, that ancestor that was engulfed. So it would be hard to find the exact modern-day example that we could use experimentally to demonstrate the same thing. And then what happened to the cell wall? So that disappeared at some point. So what is that process by which you can endosymbiose something and then reject parts of it and establish a permanent arrangement? And that's important to know, because when you understand this, then you understand a little bit better how perhaps other things that we look at inside of a eukaryotic cross-section of a cell, we say, well, that's the nucleus, that's the Golgi, that's endoplasmic reticulum. Where did those all come from? And it's possible that things like the nucleus, which also has a double membrane, could actually be microbial in origin and be simply the result of something eating something. And maybe the endoplasmic reticulum, same kind of thing happened. So we study the organelles to see if it tells us even more about our own cells than we would have thought otherwise. The origin of our cells and their structures. So it's kind of exciting. I find organelles really exciting. Now we go to the interactions. Here's where we got to be a little bit careful. Some of the pictures and concepts will be a little bit hard to look at. But I hope that at the end of the day, the take-home message will not be that eukaryotic microbes are disgusting. And I'll try to end on that strong, that we depend on them for so many other things. But I'm going to show you a few examples that have had major societal, economic, humanitarian impacts on human populations. Here's a couple of just quick examples of eukaryotes, protozoa specifically, that can cause human diseases. So you know malaria, for example. This is a eukaryotic microbe. African sleeping sickness, which is carried by the tsetse fly. This is also a protozoal infection. And the tricky thing about these types of pathogens and their infections is that the cells that are infecting the human cells in these cases are a lot like the human cells in terms of their makeup, their structure. So it's very hard to treat these infections. Unlike bacterial infections, give antibiotics, no problem. They don't affect us or amoeba. But in this case, if you have something that's anti-malarial, it may actually be anti-human as well. And the trick is to kill the malaria before it kills the human. And so that's one of the real challenges of treating diseases like this. Okay, so these are a couple of examples: malaria, African sleeping sickness. And then there's also other types of fungal infections. The last two were protozoal examples. But fungal infections can also be a problem. And I would argue not so much for those who have very strong immune systems, but for those who are immunocompromised, like the very young, the very old, or HIV patients, for example. Some of these fungal infections can be particularly problematic, like oral thrush, for example, which is caused by a yeast, Candida albicans. And you can see here this growth of Candida on the tongue. It grows on the sides of the mouth as well. And you'd think you could just go in and scrape it off, physically remove the infection. Leave your tissue raw and bleeding, and so it's not an easy thing to do to treat, and it's extremely painful. So oral thrush, major problem for immunocompromised individuals. Part of the reason why when I take my kids to the swimming lessons, I can't stand walking through that changing room, wet floor, because I know that's a really good location to spread fungal infections like athlete's foot, for example. So that's a fungal infection, one that may be easier to treat because you can put ointment right on it. So athlete's foot also caused by fungal microbes. You'll be glad to know that for these two slides here, I'll be very clear with you. I'm not going to ask you to remember the exact genus species name and what diseases they're causing on these two slides. Although I want you to know that malaria and African sleeping sickness are protozoal infection, and we got athlete's foot and oral thrush. This is fungal infection. I get that. But the next examples I'm going to give, I do want you to remember 3 genus names and what they cause specifically. So I'm going to spend a little bit more time on them. Yes, arrows, that's obvious. Oh, so in case it wasn't obvious, this is this, and this is this. Okay, photomicrographs, and then their infections. So this is number 1, and this is a big one because this is a fungal infection of plants, and it's a serious problem to tomatoes and potatoes, which may be okay to, and by the way, this is a really unusual organism. I call it a fungus, but it's a really unusual fungus. So Phytophthora, so that's the name there I want you to remember, the genus name, Phytophthora, which is, yes, a fungus, but it has some features a little bit like algae. So it has cellulose cell walls, which is kind of unusual in a fungus. And so maybe that's part of its adaptation to plants, and part of that mechanism of infection of plants look a little bit like plants. It's a really unusual fungus, but it infects tomatoes and potatoes. And it is a serious problem if, for example, you have a country where 1/3 of your population, that's all you eat, potatoes. You grow potatoes, you eat potatoes, and you have one type of potato as well. And if this happens, like in Ireland in the 1800s, for example, then you're going to have a problem feeding people. And that is the cause, Phytophthora is the cause of this great Irish famine in the mid-1800s, where you have literally 1 million people dying and dead of starvation because your potatoes look like this and you can't really eat them. And as a result of that, not only do you have 1 million people dying because you can't eat your main food crop, but you have another 1 million people that have to go somewhere else. You have to leave the country, you go to New York, you go to Canada. Massive economic impact and massive biogeographical impact on the human population as a result of one fungus and one crop. And so it's very sad, and of course there's kids involved and there's elderly involved. And if you could imagine your family trying to deal with this, there is no food. It's that kind of a situation faced by millions of people in Ireland at that time. So it's hard to imagine what that would have been like. Oh yeah, and then there's my walk to work. So that's one, Phytophthora, Irish potato famine. And then my walk to work is another beautiful thing to do in the fall. We have all these changing colors. You see it all around you. And yet you look carefully as you walk and you realize not everything's quite right. And one example of where not everything's quite right is as you pass underneath the countless maple trees that you see everywhere around you. At this time of year in particular, there is a fungal parasitic infection which is common in our region specifically. It's called Rhytisma. And that's another genus name that you should remember. Rhytisma, which specifically refers to the tar spot that you see on these leaves right here. So Rhytisma in Greek means a patch, easy to remember. And so look at the leaves, see them, and notice that these leaves here on the maple tree in front of my house have these black spots on them. And you may have seen them before and kind of ignored them, but now you can't anymore. From now on, you'll know exactly what Rhytisma is, and you'll know that it's a fungal parasite. And I'll show you. In the past, I've actually handed these around the class so you can see them up close, but in M3, it is way too big to do that successfully. But that doesn't mean you're off the hook. I'll explain in a second. So there's maple leaves with the black spot, and there's sort of two types of maple tree here, and you can see that they are really nice and dark and actually bent a little bit, so it actually bends the structure of the leaf. And you can see on another example here I have, which shows that there's actually an impact on the photosynthesis capacity of the leaf itself, because you have 3 spots of Rhytisma here, and then the rest of the leaf has begun to decompose here. So there's no more photosynthesis returning to the plant. So it's a parasitic relationship. But it's one that's remarkably efficient because the parasite, Rhytisma fungus, knows not to kill the host, or else it has no more place to live. And so what it does is at the end of the summer, when there's not too much damage done to the tree, it begins to establish this growth and then the fruiting body structures, the spores, the black material here, that once these leaves fall off the tree, will stay underneath the snow until the spring, and then it starts raining in the spring and those spores get dispersed back into the new leaves on the new maple tree buds and begin the infection anew. And you see it in the fall coming out as these nice fruiting body spore structures. The neat thing about it is the reason why I said it, you find them in Waterloo, for example. You know when you see these on the trees that you're living in a location where you have a low amount of air pollution, because Rhytisma does not like sulfur dioxide. So if you go to Toronto, look for maple trees, see if you can find Rhytisma. I'd be curious if you can. Find it here, it's a good sign. I don't know if you'd find it in Toronto. So that's Rhytisma, tar spot. You find it all on the way to work. And some of the maple trees have it, like this one right here, has lots and lots of tar spots. And if you look beside it here, there's another species of maple that doesn't seem to be affected at all by it. So some types of maples are not affected by it at all.
Welcome to my backyard. I wanted to show you what Rhytisma looks like up close and personal. And so here is right behind me signs of fall, and I've got a tree, maple tree right behind me. We were just talking about Rhytisma, and this is a fungal infection of deciduous trees. And Rhytisma acerinum is the particular species that infects maple leaves. And so I'll show you here just by walking by behind me. I'll pick up a maple leaf to show you what the infection looks like. So You'll see now this is why they call this disease tar spot, because you can get an idea here of just how black the infection looks like here. So this particular spot here will release spores, and it will release spores particularly in the spring when it's raining. All these spores that are released will end up being thrown back into the tree, into the new leaves that are forming in the spring, beginning the infection cycle anew. And so really by preventing these leaves from falling and sitting there through the winter to then distribute the spores in the spring, that's the best way to reduce the infection of this parasitic fungus. It is parasitic on the tree, but it is largely cosmetic. It doesn't seem to do too much long-term harm to the tree itself. One thing you will notice though, take a look in the fall, when you find tar spot disease, you won't notice this as much in the summer because the entire leaf will be largely green, but here in the fall most of the leaf has gone yellow except for the region of the leaf closest to the tar spot. It still has some chlorophyll remaining in it, and so it appears as though perhaps this fungus is able to influence the leaf to continue producing chlorophyll and photosynthates for it to help sustain the infection. So that's one possibility, and something you might observe and look at as you get up close and personal with this infection. And this is a really good example of how we as citizens can interact very closely with microbiology. And so this particular infection by a fungus is harmless to humans, so you can pick these leaves, talk to your friends about rhytisma, show them what microbiology looks like here up close and personal in Canadian summers and falls through the tar spot infection. So that'll give you something to look for on walks in the woods or on your street from now on. And so there you go, microbiology up close and personal for you. And I'll continue by showing you a few other pictures of rhytisma infecting maple leaves in particular in the Waterloo region.
Oh, and then there's, so that's number 2. Phytophthora for the potato blight, rhytisma for the maple trees. And now here's one, of course, that you will never forget as much as you try to, and this is where I kind of give you a little warning. This is cordyceps. Okay, I'm better than David Attenborough, so I'll do the voiceover if it's okay with you. The jungles, this is in Asia, host 80% of the insects in the world, so it's a hot spot for insects, especially ants, like these bull ants right here, that make their way along the forest floor, and when they do, occasionally they pick up spores of the cordyceps fungus, and when they do, they behave very strangely. Because what happens is the spores, the fungus, burrows its way with pressure and hydrolytic enzymes through the cuticle of the bull ants and get inside and begin to digest the tissues and infiltrate the brain of the ant. So as this process is going on, digesting the tissues into the brain, the ants behave really strange. And they start to climb. That's what happens when it reaches the brain. They start to climb. They have that one impulse, get up as high as I can, above the colony, and clamp down with my mandibles, and that's the last thing I'm ever going to do. Clamp down. Now other ants can spot this behavior. They know there's something wrong, and they take that infected ant, they run with it as far as they can, and they chuck it out of the colony. Because if they don't, that clamped down ant will be done, and the fungus will then exit the body of the insect, the ant. From the back here, you can see we have a fruiting body of the fungus. The fungus has run out of food, has digested everything on the inside. It's time to move out. And this fruiting body takes about 3 weeks to grow to full length. 3 weeks, so this is stop motion animation, or photography here. And the spores will form at the very end of this fruiting body. And as soon as that happens, and it rains or there's wind, those spores will then land on all the other ants below, and this infection in this way can wipe out an entire colony in a very short period of time, and leaving these ants as essentially shells of themselves with fruiting bodies exiting in weird and wonderful ways. There are thousands of species of cordyceps fungi, and each one of them is specific to a different species of arthropod, so different insects can be infected in the same way. And even the name cordyceps is referring to the club head-like structure of the fruiting body. And so this is, I guess, what used to be a grasshopper with the cordyceps fruiting bodies coming out of them. And some other examples coming up as well. So it's a fantastic way to distribute those spores. You would not see this commonly on your way to work like you would see Rhytisma, for example, but that doesn't mean that they're not common here. You can probably find examples of them if you looked hard enough here, because again, these are very specific, thousands of types, each one unique to a certain species of arthropod. And I think they're really spectacular. There's a video game based on this kind of a concept, isn't there? Does anyone know what it's called? The Last of Us? And that's where a cordyceps evolves to be able to affect humans? Okay. Yeah, it's cool, and leaves these ants looking weird and wonderful, fruiting bodies. Cordyceps, if that's how you spell it. Cordyceps. Are some of you feeling a little bit disgusted by this? If that's the case, then you're going to have troubles with this, because this used to be a spider. And so you can see just how beautiful these fruiting bodies can be to this cordyceps species that affects that type of spider. And if you find that disgusting, I think it's about to get a little bit worse, because, well, actually, before it gets worse, it'll get better. This is a video here, which I should just start, to show you that it's not always seen as disgusting at all. In fact, it can have a major economic benefit as well. And this is in Tibet, where there are individuals who are employed full-time with the task of finding cordyceps fungi infecting caterpillars. And so this is a caterpillar that he's just discovered with a cordyceps fruiting body sticking out the top of it. He's got the whole thing. You give it a little clean, give it a little brush off, and you take it to market. You take it to market, and the reason why this is a useful thing to do is because each one is worth its weight in gold, and it's sold for its medicinal properties, pharmaceutical properties. It's thought to maybe be one of those aphrodisiac-like effects, or give you strength or energy. So much so that, you know, get used to the idea here. I'm not going to do it. But if I were to ask you, how many of you have eaten cordyceps before? There'd be a large number of hands that would go up in this room. And it so happens that these cordyceps here, these caterpillars, with the cordyceps fungi, this fruiting body coming out the top, so remember the inside's already digested with fungus, and the fruiting body has come out the top. These are what are sold and packaged for consumption. And last year, when I mentioned this, it took a few hours before I suddenly got a tweet. A tweet from Patrick Lynn, I'm sorry. And Patrick said, had some cordyceps today. So I wrote back and said, what are you talking about? He said, well, my grandmother makes a soup with cordyceps in it. And this is the soup, this is his spoon, he's eating them. And the idea is that his grandmother tells him, and his mother was telling him, that it makes you healthy, makes you strong. And so many consume cordyceps as a food source, and I'm fascinated by this. I would love to try it. If any of you have grandmothers or mothers that make you cordyceps soup, can you save some for me and reheat? Yes, you do? So the question is, are they soft or crunchy when they're in the soup? Kind of crunchy? That's what I thought, because Patrick was nice enough, or Patrick's grandmother and mother were nice enough last year when I expressed extreme interest in this, to provide me with cordyceps, which I then placed in ethanol, probably unnecessarily, but at least they'll last forever. And so I have some here, and for those of you who are really fascinated by this and interested in it, and who don't have that experience of eating them regularly, for example, then please come take a look. And I'm also going to throw it out there that I'm looking for a source. I'm looking for maybe a few more. I have reasons for this. No, I should be very careful. They're not the reasons you just thought of. But I am actually curious to know, A, what they taste like, and I also like to occasionally give them out to people when people show an interest in them in particular. So I'd love to have a little source of them, because I find this fascinating, and it does capture the imagination of students, and it's a little bit icky at first, but get used to it. People eat different things, and so it's just the way it works. All right, so that was gross, a little bit gross. That's the worst of it for now, because this is what I want you to remember. I know it won't work, but I'm going to do my best to say, this is what eukaryotic microbes do. Every second breath we take is thanks to eukaryotic microbes, right? Those that make oxygen in the ocean, the algae, for example. So we have to be thankful for their contributions to both providing energy and oxygen, so primary production in the oceans, biomass for others to consume. We have to appreciate that if we go walking in that forest on one of those dates, you can point out that you notice there aren't carcasses all around you. There should be by rights. There's so many animals. They're all dying. Where do the carcasses go? Well, within 1 to 3 days after death, the things are gone, largely decomposed by eukaryotic microbes. So fungal degraders, protozoal degraders are huge recyclers of nutrients. And some of them as well, this is really important, this #1 most abundant carbon source on the planet, cellulose, has to get broken down somehow. And the eukaryotic microbes are the champions in doing this. Here's one example here. Remember the termite guts? Well, here's what they look like. Here's termite gut symbionts that do this very process of breaking down cellulose. So these are the protozoa that are found inside of termite guts. I show the video because these cilia here, just beautiful, undulating, flowing cilia patterns here in these termite anaerobic protozoa symbionts. And you know there are other organisms inside of there that are helping them with that process, right? Inside of the termite guts, methanogens, inside. So that's what they're doing, and that's the value of eukaryotic microbes, recyclers of nutrients, degrading plant matter, primary producers, oxygen producers, the vast majority of them. There are a few bad apples, like the Ebola and the virus world for humans. There are a few bad apples in here that can also infect humans or other organisms. And that's it. We're done. Eukaryotes, done. So, let's jump to viruses for a few minutes. And then I've got a couple of clicker questions to test on you. So we're moving over into, again now, the viruses. We're only going to do a little history. Because, as you can imagine, in a world dominated by the thinking of Louis Pasteur and others at the time, contemporaries, germ theory of disease was just establishing itself. How complicated is it when you know that the causative agent of disease is so small, it passes through the filters that should be holding back the bacteria? So now you have another entity, you have another infectious agent, really hard to figure out because you can't grow them like you grow bacteria on plates in 240L. So let's start with the history, and next class we'll do the rest, or some of the rest. So viruses have been around with humans for a long time. And you can see it if you look at these mummies of Pharaoh Ramses the whatever here in this picture. You see examples here of the skin on the face having signs of damage to it, which is an indication that this particular pharaoh would have succumbed to smallpox infection, which is a viral infection. And so at least in ancient Egypt, it was apparent that viruses were still around with humans, which is not saying very much, because viruses likely have been around a lot longer than that. Geological time we're talking about. Okay, so they were probably around as prokaryotes evolved across geological time, infecting bacteria and archaea for millions or billions of years before we even showed up. So our interaction with viruses is relatively recent in viral history. So in the 1800s, we began to figure it out, and the first time it was figured out that there was something really small infecting something else was with this tobacco mosaic virus, which leaves tobacco leaves wrinkled and with a pattern on it. And the infectious agent, it turned out, could be filtered and passed through where the bacteria would have been held back. So the infectious agent went through that little filter. And so that was Dmitry Ivanovsky, who said, must be something else, some other type of particle, and was really the first to identify an infectious disease caused by viruses. And by the way, I usually say with the bacteria that there's a few bad apples that do us harm, and with the archaea, well, there aren't any, and in the fungus, there are a few bad apples again. But the viruses, they're all bad apples, every single one of them. Not to humans, they're not all going to infect us, because we're covered in them, we're fine. But to the bacteria and the archaea and the fungus and all other types of organisms on the planet, of which we're not very important players, the viruses are always bad news. So they always infect, they always cause lysis and disruption to the cell. They're software that don't run very nicely. They don't play nice with the operating systems of life. So Dmitry Ivanovsky figured it out, small things passing through, so they're not bacteria, and that's the first time viruses were discovered. And then the history takes a turn to Quebec, to Canada. So I start to get proud, and heck, we're talking about Quebec, so maybe we say go Habs go tonight. No, that didn't go over well. I'm born in Montreal, so you gotta excuse that comment. But I'm also diplomatic, so I hope that the best team wins tonight. It'll be exciting to watch, and I'll be watching. But Félix Derel was a French-Canadian who discovered bacteriophages, those viruses that infect bacteria. And what he realized was that if you had a culture of bacteria, sometimes the culture would go clear overnight. Something just cleared all those cells, lysed them all overnight. So he knew that happened. And he also knew that if you take bacteria and you spread them out over a surface of a plate and let them grow, so you get a lawn, which is what's shown on these plates here, from that clearing fluid, add that to the plate, and you end up with all these circles, these clearings in the colony. So something would lyse and disrupt those cells in a ever-expanding concentric circle. These little circles be referred to as plaques, and they're still called plaques, by the way. And the reason why they're called plaques is in those old buildings that you still see. Go to Europe, or I guess Quebec as well, you'll see on these historical buildings you have these metal plates, these circles, these plaques on the front of those buildings, and look a little bit like the plaques. So that's what they're still called. And so Félix Derel discovered viruses that infect bacteria in this way. And then sort of the last bit of history is this Walter Reed, who was a bacteriologist and what's the word for them? Pathologist, working with the US Army around 1900. And there was a problem. Yellow fever was a huge problem. People were getting sick, people were dying. About half that get it die. It's got a kill rate like Ebola, for example, right now. So yellow fever was a big problem, and part of the problem is how do you stop it and treat it if you have no idea what causes it, or importantly, how it spreads from one person to another. Some people thought it was through touch, and other people thought it was through drinking water, but that wasn't consistent, it wasn't adding up. So what Walter Reed was convinced, he had a hypothesis, it's the mosquitoes that are a vector for whatever this yellow fever is, it's a virus. So he was doing a lot of work that was completely unethical. And so he set up experiments, and he had release forms that he would offer to the soldiers, and he'd say, look, you want to be a part of this experiment? I'll give you $100. If you get yellow fever during the experiment, of which you probably will, you get an extra $100. And that can go to your family, you can send it right off to your family. So people were signing up. And he then had people that were exposed to insects, the mosquitoes, who allowed them to bite. They would get sick, and so he was trying to demonstrate through these robust experiments, very good scientist, terrible ethics policy, never would work today. And he would even do it to himself. So he'd take an insect, put it in a tube, stick it on him, let it feed, and sadly it didn't work with himself. So he was really, that didn't go over so well. In the end, the experiments themselves were robust enough that it was clear that mosquitoes were the vector. That was a huge step towards figuring out, don't go and walking in the swamps at night as some of the soldiers were doing. You're just going to get yellow fever. And so it saved many, many lives, this research. And at the end of the day, he got his wish. He contracted yellow fever and he died. So it sort of has a happy ending in a weird, twisted way. So his team was also responsible for figuring out that it was a virus particle as well. So they identified that at the time. So they did a lot of good work towards understanding this huge problem where the largely men soldiers were working at the time. So, Walter Reed. Very quickly, they're all intracellular obligate parasites, 10 and 100 nanometers. So we're talking about maybe a tenth of a micron, which is a tenth of a bacterial cell, or smaller than that. So here's examples of really small virus particles. Here's T4 bacteriophage that infects E. coli, right here. We'll come back to T4 bacteriophage soon. And they have really small genomes of a few thousand bases to 200,000 bases. I'll give you a little trick here. When you hear that, a gene's 1,000 bases. Think of that, on average, 1,000 bases a gene. So you know they have a a few genes to maybe 200 genes most of the time. So that's the kind of genome they have. Pretty small, not too many genes. And yet, and this is one where you're going to have to use judgment, because I'm not necessarily going to say, tell me the exact genome size of these viruses here. These are those exceptions to the rule again, that there are some viruses that are way off the charts, much bigger than you'd expect from the norm, which is shown here in relation to E. coli or a human liver cell, which is much bigger than E. coli. Here's the exceptions. Good name. Megavirus, found off the coast of Chile. A double-stranded DNA virus. Has a really big genome, getting close to bacterial genomes in size. So 1,200 genes here. 1,200 proteins. And then another one was discovered called, and I never would have called it this, called the Mimi virus. That doesn't sound very big or tough, but that was what was intended to be communicated with the naming of the Mimi virus. And it also has large genome, bunch of proteins, but nothing compared to the Pandora virus, which was discovered very recently. And you can see it right here in relation to the Mimi virus and a bacterial cell here. These are really big viruses, and it challenges our concept of what a virus actually is. If it's bigger than some bacterial cells, that's really bizarre. So the Pandora virus, just found in 2013, published in Science. Big genome, the average size of a bacterial genome is exactly this, 2.5 megabases. So it's got a bacterial-sized genome. It's a micron in diameter, so it's the size of E. coli. And all of these, you probably may notice this because it's kind of curious. What's common to all of these giant viruses? Probably a better take-home message than the actual names of these. Where do you find them? All three. In amoeba. Amoeba, amoeba, amoeba. These protozoa with the pseudopods that move in amoeboid manners. That's where you find them all. And this is where you find the Pandora virus. It's one of the, you ever heard of Pandora's box, and you open it up and all these things come out of it. Big surprise. So here they are inside of an amoeba cell. You look at them and you think they were bacterial cells or inclusion bodies, carbon storage. Turns out they're viruses inside of amoeba. So amoeba keeps coming up here as accumulating things inside. We've had a few examples already, Legionella. Now we have giant viruses. Amoeba are kind of neat.